Hello, everyone. Welcome to our lecture number 5 in our authoritative series on the chemistry of carbohydrates. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. Having thoroughly covered monosaccharides, disaccharides, glycosidic bonds, digestion, and lactose intolerance in the previous lectures, today we move to the higher order carbohydrates, oligosaccharides, and the polysaccharides. These molecules are central to cell recognition, structural integrity, energy storage, and numerous pathological processes. Mastering them is essential for understanding glycoproteins, blood group antigens, glycogen storage diseases, and connective tissue disorders. Let us proceed with the precision and clinical correlation. Let's start with the definition and attachment of the oligosaccharides. Oligosaccharides consist of 3 to 10 or sometimes more than 10 monosaccharide subunits linked by glycosidic bonds. They are rarely free in nature. Instead, they are almost always covalently attached to the proteins to form glycoproteins or the lipids to form glycolipids. Two major types of attachments exist in oligosaccharides. The very first one is the N-linked. The N-link attached to the nitrogen atom of the asparagine residue. And the second one is the O-linked. O-linked attached to the oxygen atom of serine or threonine or to the hydroxyl group on lipids. This glycosylation is not random. It is highly regulated and plays critical role in protein folding or protein folding stability, trafficking, and biological recognition. O-linked oligosaccharides are attached via glycosidic bond between a sugar residue and the hydroxyl group of the serine and threonine on the protein backbone. These chains are greatly in complexity and length. They are heavily involved in cell-to-cell recognition and interaction, modulation of enzyme activity, protection of proteins from the degradation. O-linked glycosylation is particularly abundant in mucins, glycoproteins that form mucus, and many membrane proteins. What is N-acetyl-glucosamine? A particularly important and dynamic form of O-linked glycosylation is the attachment of a single N-acetyl-glucosamine to serine or threonine residues. This reversible modification, known as O-N-acetyl-glucosamine or glycosylation, right? Regulates numerous cellular proteins, including enzymes and transcription factors. Interestingly, this modification often alternates with phosphorylation at the same site, exerting opposite regulatory effects. One may activate while the other inhibits the protein. This yin-yang relationship is a key mechanism in cellular signaling and is increasingly linked to diabetes, neurodegeneration, and cancers. N-linked oligosaccharides are usually more complex and branched than O-linked ones. The process begins with the attachment of N-acetyl-glucosamine to the amide nitrogen of asparagine within a specific 3 amino acid consensus sequence, that is asparagine or the X or the serine or the threonine. Subsequent addition and trimming of sugars by glycotransferases and glycosidases in the endoplasmic reticulum and Golgi operators produce characteristic branch structures. These complex N-glycans are crucial for proper protein folding, quality control, and recognition by lecithins or lectins. What about the examples of oligosaccharides? Common oligosaccharides include trisaccharides, that is raffinose, that is the combination of glucose, galactose, and fructose. Next is the tetrasaccharide, that is stachyose, two galactose molecules, one glucose and one fructose is in its structure. The next one is pentasaccharide, that is verbasacose, right? Three galactose, one glucose, and one fructose molecule is present in its structure. The next is hexasaccharide, that is aigose, that contains the four galactose, glucose, and fructose molecule, right, in its structure. These are found in legumes and are poorly digested by humans, often causing flatulence due to bacterial fermentation in the colon. Next is the polysaccharides. Let's talk about its classification. Polysaccharides means more than 10 sugars are present in its structure. Polysaccharides are further divided into homopolysaccharides and the heteropolysaccharides. Homopolysaccharides made up of one type of monosaccharides. For example, the starch, glycogen, dextrin, dextran, aniline, and cellulose. And what about the heteropolysaccharides? Contain two or more different monosaccharide units present in its structure. The examples are glycosamine glycans, the GAGs, mucilages, gums, and pectins, right? Other may include the glycoconjugates, include proteoglycans, glycoproteins, the glycolipids. This classification helps in understanding their diverse biological roles, from energy storage to structural support and lubrication. What about the general characteristics of polysaccharides? Polysaccharides are high molecular weight polymers, often exceeding 200,000 Daltons, right? They are typically with amorphous, tasteless, and non-reducing because their anomeric carbons are involved in glycosidic linkage. In aqueous solution, they form colloidal suspensions rather than true solutions. These properties make them ideal for long-term energy store in case of the starch and glycogen, and structural function, the cellulose. Let's talk about the starch, the major plant storage form of polysaccharides. Starch is the most abundant storage polysaccharide in the plants. It is a mixture of two forms. The very first one is the amylose, and the second one is the amylopectin. Amylose, 10 to 30% is the contribution in the starch is by the amylose. That is unbranched polymer of glucose linked by alpha 1,4 glycosidic linkages. This is very important in exam point of view and in a viva point of view, right? So this is an unbranched polymer of glucose linked by alpha 1,4 linkages, right? Or glycosidic bonds. Next is the amylopectin, 70 to 90%. Highly branched polymer with alpha 1,4 chains and 1,6 alpha 1,6 branch points every 12 to 30 residues. The molecular weight ranges from several thousand to 500,000 Daltons. Starch is the primary dietary source of carbohydrates for the humans. As you can see on your screen, the amylose is basically an unbranched polymer of the glucose linked by alpha 1,4 glycosidic bonds, and the amylopectin is a highly branched polymer with the alpha 1,4 chains and alpha 1,6 branch points every 12 to 30 residues. Amylose adopts a helical conformation in water. Iron molecules can insert into the center of this helix, producing the characteristic blue color that is a diagnostic for starch, while the amylopectin, being branched, gives a reddish purple color with iodine. This simple test remains useful in both lab or laboratory and clinical setting for detecting starch molecules, and this test is known as the iodine test. Next, let's discuss the dextrins. Dextrins are intermediate products formed during the partial hydrolysis of the starch by acids or amylase or the alpha amylases. So, dextrins are the products of the amylases enzyme that can be obtained from the starch molecules, right? And this is one of the polymer or the polysaccharide, right? So they consist of a complex mixture of glucose molecules of varying sizes, and contain mostly alpha 1,4 linkages with some 1,6 branches, right? Or alpha 1,6 linkage at the branch point. So dextrins have a mildly sweet taste and used as mucilages, glues in infants formula or infants feeding formulas, and in starch hydrolysates. They are classified as amylodextrins, erythrodextrins, or acrodextrins based on their reactions with the iodines. What about dextrans? Dextrans are branch polysaccharides produced by the bacterium through the action of transglucosidases on the glucose molecules, right? They contain alpha 1,6, alpha 1,4, and alpha 1,3 linkages. Clinically, or clinical dextrans have molecular weight of 40,000, 70,000, or 75,000 Daltons and are used as plasma volume expanders in the treatment of hypovolemic shock. They improve blood volume without the risk associated with the blood transfusion. So, thank you very much for your attention. We have now completed a detailed and authoritative study of oligosaccharides and polysaccharides. You have learned how oligosaccharides function in protein glycosylation and or O type of glycosylation, their regulatory roles and their structural properties, and clinical uses of the key polysaccharides such as starch, dextrins, and dextrans. These concepts form the bridge between carbohydrate chemistry and critical area of medicine, including immunology, hematology, gastroenterology, and metabolic disorders. In the next lecture, we will explore glycosamine glycans, proteoglycans, and mucopolysaccharides or mucopolysaccharidosis, topics of immense clinical importance. Thank you very much for watching lecture number 5 of this series. If this lecture has strengthened your understanding of higher carbohydrates, please like the video, share it with your medical fellow students and juniors, and subscribe to the channel for more professional, high yield, and exam oriented medical biochemistry lectures. Turn on the notification bell so you never miss the upcoming content. Please post your question or doubts in the comment section. I read every single comment and will respond personally. Which aspect of oligosaccharides or polysaccharide would you like me to explain in greater detail? This is Dr. Muzamil Liaqat Ali signing off. Continue to revise the structures of amylose, amylopectin, and types of glycosylation daily. Stay focused, stay consistent, and keep learning the biochemistry with the passion and scientific rigor. I look forward to seeing you in the lecture number 6. Thank you once again.